Hello everyone. The topic for my today's presentation is biosafety concern of transgenic plant. So this is the outline for my presentation. Introduction, transgenic plant, the biosafety levels, National Biosafety Regulatory Framework in India, what are biosafety concern, risk to human health, environmental concerns, ecological concerns, socio-economic and ethical consideration, conclusion and references. Let us start with the introduction. Basically, what are transgenic plant? A transgenic plant contains a gene or gene of different species artificially inserted in its genome. The inserted gene sequence is known as transgene that can come from unrelated plant or from a completely different species. The transgenic crops are important as conventional plant breeding involve exchange of gene between plant of the same species or from closely related plant to produce a hybrid having desired trait. The useful gene can be introduced not just from within the crop species or from closely related plants, but even from a wide range of other organisms. Transgenic crops are produced through genetic engineering in which genes that code for desirable trait are transferred from one organism to another. The biosafety refer to the policy and procedure adopted to ensure environmental safety during the course of development and commercialization of genetically modified organisms, which we also known as GMOs. Now let us see how these transgenic plants are prepared. Genetic engineering begins with the identification of the gene responsible for a trait of interest. Restriction enzyme recognizes and cut the DNA at specific region, and the other enzyme known as ligase joins the end of that 2 DNA strand. One of the gene has been isolated and cloned. It has to undergo several modifications before it can be inserted into plant. Identification and locating gene for agriculture important trait is currently a limited step. Once a gene has been isolated and cloned, it must undergo several modifications before it can be effectively inserted. So in these, it involves two type of steps. The first involves a device called as gene gun. The DNA to be introduced into the plant cell is coated onto a tiny particle. These particles are then physically shot onto the plant cell, and some of these DNA come off and get incorporated into the DNA of the recipient plant. Here we can see the gene replication take place. It is coated with the gold particles. The cell is shot and the DNA incorporate in the plant cell chromosome. The second method uses the bacterium Agrobacterium tumefacium to introduce the gene of interest into the plant DNA. Here we can see this is the gene of the interest which is inserted into the TI plasmid. This bacterium is mixed with that plant cell, and after that the TI plasmid moves into the plant cell and insert the DNA into the plant chromosome. After these two steps, after that we can just screen for the cells which have transgene. Here we can see the cell which is having the transgene transformed cell are selected with the selectable marker, and these transgenic plant regenerate from the single transformed cell. Likewise, we can produce the transgenic plant. Now let us see what is biosafety level. These are the combination of laboratory practices and techniques, safety equipments and laboratory facilities appropriate for the operation performed and the hazard posed by the infectious agent. The proposed safety levels for work with recombination DNA technique take into consideration the source of the donor DNA and its disease potential. There are 4 biosafety level. Here we can see biosafety level 1, biosafety level 2, biosafety level 3, and biosafety level 4. What is biosafety level 1? It is appropriate for undergraduate and secondary education training and teaching laboratory and for other facilities in which work is done with defined and characterized strain of viable microorganisms which do not cause diseases in the healthy adult human. In the second one, biosafety level 2, it is applicable in clinical, diagnostic, teaching and other facility in which work is done with the broad spectrum of moderate risk reagent. Laboratory workers are required to have specific training in handling the pathogenic agents in this type. The next one is biosafety level 3. It is applicable to clinical diagnostic teaching, research or producing facilities in which work is done with indigenous or exotic agents where the potential for infection by aerosol is real and the disease may have serious or lethal consequences. The next one is biosafety level 4. It is applicable to work with dangerous and exotic agents which pose a high individual risk of life-threatening diseases. The strict training and supervision is required and the work is done in specially designed lab for this kind. National Biosafety Regulatory Framework in India. Government of India have evolved a comprehensive regulatory mechanism for the development and evaluation of the GMOs and rDNA work. The Ministry of Environment and Forest is the nodal agency for release of GMOs in the country, and the minister has enacted Environment and Protection Act 1986 to provide the protection and improvement of the environment and the related matter. The rules and regulation cover the area of research as well as the large-scale application of GMOs and products made from them. There are 6 component authorities for the regulatory mechanism as described here. The first one is Recombinant DNA Advisory Committee. This is a committee constituted by DBT referred to as RDAC, which take note of the development in biotechnology at national and international level and recommend safety regulation for research and application. The next is Institutional Biosafety Committee. It is a nodal point for interaction within the institute for implementation of the guideline. For these institution carrying out research activity on genetic manipulation should constitute IBSC with 1 DBT nominee. The next is Review Committee on Genetic Manipulation. DBT has next high level of body known as RCGM, which has the functions to bring out manual of guidelines, specific procedures for regulatory process of GMOs in research, use and its application in industries. The next one is Genetic Engineering Approval Committee. It functions as a body under the MOEF and is responsible for the approval of activities involving large-scale use of hazardous microorganisms and recombinant products in research. The next is State Biotechnology Coordination Committee. In each state, there is a State Biotechnology Coordination Committee headed by a Chief Secretary where research and application of GMOs are done. The next one is District Level Committee. DLC is the District Level Committee headed by District Collector as an individual to monitor the safety regulation. Both SBCC and DLC work together with RCGM in the inspection and monitoring of the experiment at the field site. Now let us see what are biosafety concerns. The transgenic crops are released for field testing and commercialization. Concerns have been expressed regarding potential risks to both human and environment. Biosafety describes the principal procedure and the policies to be adopted to ensure the environmental and personal safety. Recognizing the need of biosafety in genetically engineered crop and the development of the activities, an international multilateral agreement on biosafety CPB is done in 2003, whose main objective was to set up the procedure for safe transboundary movement of living modified organism and principle and methodology for risk assessment and establishment for information sharing through the biosafety clearing house. Now let us see the biosafety concerns or risks which are related to human health. The first one is risk of allergies. Basically, we know that risk production of GMOs sometimes include the introduction of new protein from the organism which have not been consumed as food and may cause allergy. All known food allergies are proteinaceous in nature, mostly heat stable and resistant to proteolytic digestion. But most protein do not show any allergic effect on human. It may be noted that the allergies can be developed by any individual to even common food and eggs, fish, etc. But WHO have concluded that GM food have the potential to cause allergic reaction, but this risk is comparatively low to the risk comparatively high than that of traditionally grown food. The next one is risk of toxicity, which can be caused to human. It can be done as the risk of toxicity may be directly related to the nature of the product, whose synthesis is controlled by the transgene, or the change in the metabolism and the composition of the organism resulting from the gene transfer. In some cases, organism may contain inactive pathway with the addition of new genetic material which could reactivate this inactive pathway or otherwise increase the level of toxic substance within the plant. The next is antibiotic resistance. Current method of generating transgenic plant employs the selectable marker gene, which is transferred along with any other gene of interest, usually on the same DNA molecule. The use of these antibiotic resistant markers have raised the concern that eating foods carrying antibiotic resistant marker would reduce the effectiveness of antibiotic to fight disease when these antibiotics are taken with meal. The next one is eating foreign DNA. They have been dangerous for eating the foreign DNA in GM food because the piece of the DNA that did not originally occur in the plant, food plant, can be dangerous to human. Change in nutritional value. Concerns about the accidental change in the nutritional component of the transgenic crop while incorporating other traits have been raised. For example, some of the isoflavone levels in Roundup, or a controversy about the increase of the level of phytoestrogen in the herbicide tolerant soybean has been reported. The next type of biosafety concern is environmental concern. gene flow or dispersal from transgene. So basically, genetic traits with selective advantage in agriculture or natural system are liable to spread beyond the cultivated variety in which they were originally introduced. Accidental crossbreeding between the GMO plant and the traditional variety through pollen transfer can contaminate the traditional local variety with GMO gene, resulting in loss of traditional variety for the farmers. The next one is resistance or tolerance to the targeted organism. The insect population adapt to the environmental pressure poses the development of the strain which are resistant to these genes is a serious threat to a long-term efficacy of the insect-resistant transgene. The potential benefit of planting insect-resistant transgenic crop include decreased insecticide use and reduced crop damage. However, the innate ability of the insect population to rapidly adopt the environmental pressure poses the development of the strain which are resistant to these gene in a serious threat, which I have told it in long run it can affect the efficacy. The next one is generation of new live viruses or superviruses. Concern have been raised that due to GM virus resistant plant, it could encourage the viruses to grow stronger or give rise to a new or stronger variant that can infect the plant. There are two mechanisms by which this can happen. One is recombination and other is transcapsidation. In recombination, transgenics, virus recombination can occur either by copy choice or ligation. And transcaptidation is partial or fully coating of the nucleic acid of one virus with a coat protein of the different virus. Thus, in GMO, it involves the encapsulation of the genetic material of infected virus by the GMO procedure viral protein. The next one is loss of biodiversity. Cultivation of the genetically modified crop considered by some people as possible source of biodiversity loss, mainly through impact to the environment. Now let us go to the ecological concern. So the ecological concern, the first one is increased weediness. Weediness or invasiveness means the tendency of the plant to spread beyond the field where it was first planted and causing the undesirable ecological changes. This can be one of the reason. The other one is creation of superweed. GM crops may hybridize with related weedy relatives and the transgene may be transferred, resulting in the creation of superweed. The potential for a crop to hybridize with a weed is highly dependent on the compatibility and relatedness. The next one is reduction of cultivars. So these genetic erosion has occurred as the farmer have replaced the use of traditional variety with monocultures. Like in this, we know that as the traditional variety have its own characteristic, this is expected to further intensify as more and more transgenic crop are produced and introduced, which bring in considerable economic benefit to the farmer. The relative rate of the susceptibility to any of these infectious or destructive situation increases the single variety are being used. The next one is non-targeted effect. Non-target or unintended effect is another risk as transgenics growing in a particular ecosystem can cause direct or non-targeted effect on certain microbes or insect growing in a particular ecosystem. GM crop containing insect-resistant Bt toxin have been studied. Possible environmental effect includes direct effect on non-targeted insect due to exposure to GM plant material, and also any indirect effect on non-targeted species, so-called multitropic food chain, can be seen. The next one is unpredictable gene expression or transgene instability. There is considerable evidence that transgenes show instability because they become deleted or modified following their integration into the plant genome. The level of transgene expression may also decline with the passage of generation as gene slicing may occur. If and when these phenomena occur, the crop become vulnerable to such stress for which the transgene provide protection. Such changes can lead to the production of new protein that may be toxic or allergic, or may dispute or alter metabolic pathway that play a major role in making the GMO successful. Now let us go to socio-economic and ethical consideration. One of the most important consideration in commercialization of the transgenic crop is that socio-economic implication. Scientists might have come up with the techniques which may look novel or innovative, but from a global perspective may have wide range of socio-politic and socio-economic things, like the development of the genetic use restriction technology is a prominent example. The potential benefit, the other one is potential benefit to the consumer and the farmer. Due to increased seed market, the developing countries may get dependent on few suppliers. India has totally banned the use of GORT in plant variety. These called gene use restriction technology. India has totally banned the use of GORT in plant variety for registration under PPV and FRA 2001. India is a land of small farm holders, and there are about 90% of the operational farm holding in the country, and 60% of them own less than 1 hectare of the land. So farming provide a livelihood to many of the Indian population. Unless developing countries have policies in place to ensure that the small farmer have access to better agriculture input delivery system, there will be increased inequality of income and wealth for small farmer if transgenics are introduced, as big farmer are likely to capture most of the benefit through early adoption of the technique, expand production and reduce the unit cost. However, Indian farmers have adopted the biotech cotton technology in India. Another concern of developing countries is the open liberation or globalization of the market, where competition on plain level is done. These were some of the biosafety concerns which were related to the transgenic plant. This brings me to the end of my presentation. The conclusion for my presentation is, with the emerging of agriculture, biotechnological science, many genetic modified crop have developed and commercialized to feed the world. With its rapid commercialization every year, concerns are raising continuously about safety issue of GM crops on human health, food, environment, social, economical, and political. Implementing all biosafety laws, regulation, and protocol are important for safe product development and for safe utilization of these technologies. Genetically modified crop development at global level is increasing, and many GM crops are commercialized in developing countries to hasten the agricultural productivity and nutritional status of important crops. These are the references which I have used to prepare this presentation. I hope I had made myself clear. Thank you.